What's up, people? It's me, Dylan, here from C.A.R. Entertainment, and we're going to be bringing you the second video in our biology review system for AS1. We're going to start off with cells this time now. We're going to be doing microscopes, then we're going to cycle all the way back to molecules, and we're going to go through nucleic acids, proteins, lipids, etc. But I just want to get this first one out with cells and microscopes, so we'll get under our way here, shall we? So we're going to start off distinguishing between what a prokaryotic and a eukaryotic cell is. So there are two categories of cell. These are prokaryotic and eukaryotic. The prokaryotic lacks a degree of compartmentalization that the eukaryotic possesses. The prokaryotic lacks a nucleus and other membrane-bound organelles. We will get onto this. So bacteria are what prokaryotic cells are. But eukaryotic include animal, plants, and fungi cells. So prokaryotic bacteria, eukaryotic animal, plants, and fungi. Cell fractionation and organelle isolation is a technique of obtaining organelles. So cell fractionation involves homogenization and centrifugation. We want to move on now and talk about what the light microscope is like. So whenever you're viewing cells under the light or optical microscope, I'm going to refer to as the light microscope. Animal cells will appear irregular in shape, with very homogeneous, with granular cytoplasm bounded by a cell surface membrane. They have a prominent nucleus which is invariably present. Plant cells are more complex, however, as they show a cell surface membrane. They have a cytoplasm and the nucleus, a peripheral cell wall, and there's also a large central vacuole. The palisade cells in the palisade layer immediately under the upper epidermis are rich in chloroplasts. Later on we will also get into this. We're going to talk about unit of measurement because we do calculations in the exam. So just basically we're just going to start off by saying the unit of measurement is 1 millimeter, and this is 10 to the minus 3, and 1 micrometer is 10 to the minus 6. Magnification is used to calculate or estimate the size of cells and cell structures. The standard unit is a micrometer, and 1,000 micrometers is in every millimeter. So 1 million micrometers in each meter. And small cell structures, or nanometers, has 1,000 nanometers in each micrometer. You won't need to know this in depth, but it's just handy information for you to know. So magnification can be classified, can be found out by using the following equation of measured size of image divided by true size, and true size is found out by the measured size of the image divided by magnification. We're now going to move on away from the light microscope and move on to the electron microscope, or EM. So the EM uses electrons rather than light to produce these images using a high-powered microscope with resolution and not magnification. And to make this more understandable, we are going to tell you what resolution is. And resolution essentially is just the ability to see two adjacent points as distinct entities following magnification. The EM achieves meaningful magnification in excess of 1 million, and the light microscope only magnifies meaningfully up to a few thousand, and this is because electrons have a shorter wavelength than light. The cell structure is much more complex than suggested by a light microscope, as a light microscope has advantages such as it can view living specimens, but an EM can only be used in dead material because it has a vacuum inside it, and the air molecules may deflect the electron beam. Artifacts can often feature an electron micrograph image, and the specimen has to be cut very thin using specialized cutting instruments. We're now going to go on to the types of EM. So there's two types: of transmission electron microscope and the scanning electron microscope. So the TEM, which will be referred to as the transmission electron microscope, involves electrons passing through a very thin specimen with a high resolution used for a very high magnification. But the SEM, standing for scanning electron microscope, will involve electrons reflecting off the surface of the image as opposed to going through it, and this is useful for giving a 3D image of surface features, as can be seen. The differences are really what we just told you there now, with the electron microscope having a greater resolving power because the electrons have a shorter wavelength than light. But light can view living organisms and living processes such as mitosis, while the interior of the electron microscope is a vacuum, so the specimen must be dead. So there you go, guys. That's going to be the end of our microscopes video. We're going to be getting back to molecules next video. Hopefully you're enjoying these, and hopefully they are helping the shorter videos. This has been Microscopes from C.A.R. Entertainment on the AS1 Biology for the CS Spec. I've been Dylan, and I'll see you next time.